Okay, I hope you're now ready to roll up your sleeves and take a look at some dihybrid crosses. So we're going to start looking at two traits simultaneously. And this is another one of Mendel's projects. So to start out with, Mendel crossed plants that had seeds that were either yellow and smooth or green and wrinkled. So two different traits. When he did this, he got an F1 generation that were 100% yellow and smooth. So what this tells us is those traits of yellow and smooth are dominant traits, right? So we can symbolize yellow with Y, a big Y. And then that means green will be a lowercase y, little y. So that's our trait of color. And then that also means big S for smooth, and then little s for wrinkled. So this is for shape, for our two traits. Okay. So then if you cross individuals from that F1 generation, here's the phenotypic ratio that you get as a result in your F2 generation. You wind up with a ratio of 9 yellow smooth to 3 yellow wrinkled to 3 green smooth to 1 green wrinkled individual. So we're looking at a matrix of 16 possibilities. Okay, so let's walk through how to approach the genetics of this. So in our in our parental generation, we're looking at true breeding individuals. So we had yellow and smooth crossed with green and wrinkled. So big Y, big S crossed with little y, little s, right? So parent number 1 here can produce just one type of gamete, right? So only big Y and only big S. And keep in mind that all the offspring have to have a color and a shape. So both of those are going to get passed on. Same situation here for parent number 2. How many types of gametes can this parent make? 1, right? So this parent can only make little y, little s gametes. So then when fertilization happens, all of the F1 offspring are going to be big Y, big S, little s. And so now if we cross that with another individual who is big Y, big S, little s, for thinking about offspring from that cross, let's think about gametes again. Each of these individuals, each of these F1s, can produce 4 different types of gametes. And we can think of this a little bit like foiling, right? So remember that each gamete needs to have some kind of Y and some kind of S. So it's possible that a gamete could be big Y, big S, or a gamete could be big Y, little s, or a gamete could be little y, big S, or a gamete could be little y, little s. Okay? So now, if we want to look at possible combinations, so let's just write these out. So we've got big Y, big S, big Y, little s, little y, big S, little y, little s. Right? So that's one parent. Same thing for the other parent, right? So it's the same genotype. So for the second parent here, same possible combinations. Big Y, big S, big Y, little s, little y, big S, little y, little s. So, after having considered our gametes, what we've done here is we have come up with a 4x4 Punnett square. Okay, so here's my Punnett square layout. So if we want to look at the genotypes that result from this F1 cross, this is our fertilization matrix. All we need to do is combine, row 1, column 1. And as I'm doing this, so I talked previously about the convention that you always put the capital letter first. The other important convention here is to put allele pairs together. So when I'm looking at this combination of big Y, big Y, or big Y, big S with big Y, big S, for the outcome, what I'm going to write is big Y, big S, big S. And then if I go across, I'm going to have big Y, big S, little s. And I'll have big Y, big S, big S. And then I'll have big Y, big S, little s. And as a side note, it always feels a little weird to say all these bigs and littles, but it does really help in the long run for keeping track of things. I do think it's worth it at least once to do the full bookkeeping to write out all of the different genotypes that can result from this F1 cross. Okay, so here's what I just showed you, that top row. And now actually, I want you to notice something, which is that the first column is going to be symmetrical with this top row. So we're going to have big Y, big S, little s, right here. Then we're going to have big Y, little y, big S, here and here. And then we're going to have big Y, big S, little s, there. And then oh, I just noticed, you'll notice this too, I have G's instead of Y's, but the same principle applies. So just pretend that those G's are actually Y's. Okay, so then I've also filled in a couple of other genotypes here. So here's big Y, big S, little s, right? And then here also is big Y, big S, little s. And the reason I've filled all of these in is, let's just take a second and think about the phenotypes for these individuals, right? This individual has two dominant alleles, so it's going to have the dominant phenotype for both seed color and seed shape. This one's also dominant, 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 dominant. There's a trend. And if I add these up, well, we'll get to that in a minute. So if we keep going, here's what's going to show up in some of the next squares. So at this point, I've got big Y, little s, little s. Here I've got big Y, little s, little s. And then here I've also got big Y, little s, little s. And so these three individuals are dominant for color and recessive for shape. So dominant, recessive. Okay. My next individuals are little y, big S, little y, big S, little s, and then I've got little y, big S, little s. So these three individuals are recessive for color, dominant for shape. And then last but not least, down here I've got little y, little s, little s. So this individual is recessive for both color and shape. And so now, if I add these together, what we'll find is there's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 that are dominant for both traits. 3 that are dominant for color and recessive for shape, 3 that are recessive for color and dominant for shape, and then 1 that is recessive for both color and shape. Okay, so that is my how I go from my genotypes in a Punnett square to that phenotypic ratio for 2 traits. Okay, and one interesting thing here is that this phenotypic ratio indicates that there's independent assortment for color and shape. And this is known as Mendel's second principle. So it isn't as though we only get yellow smooth offspring, right? We get some yellow smooth offspring, right? We get 9 yellow smooth, but we also get some that are yellow wrinkled. And then we also get some green that are smooth, and we get some green that are wrinkled. And we can add these together in a slightly different way, and note that if we were only tracking color, we'd wind up with 12 that are yellow and 4 that are green. And we can reduce that fraction down to a 3-to-1 ratio for color. And the same is true for shape. If we add things up, we've got 12 that are smooth, we've got 4 that are wrinkled, so we've got a 3-to-1 ratio for shape as well within this overall picture, working with 2 traits. And so what I want you to think about here is that having done this dihybrid cross, that's actually the same as doing 2 sets of independent crosses. 1 cross for color and 1 cross for shape. So as I've shown you here on this slide, we can look at this in a different way and go big Y crossed with big Y little y. And hopefully by now you're getting a little bored of this monohybrid cross, and you know automatically without drawing a Punnett square that the result that you will get for your genotypic ratio is 1 big Y to 2 big Y to 1 little y. And then it's the same show if you're talking about S's, right? So you're going to get a 1 big S to 2 big S to 1 little s ratio. And now you can actually get back to that giant Punnett square if you think about things this way. You could have a big Y along with big S, big Y with big S, and big Y with little s. So the probability of having 2 big Y's and 1 big S, or 2 big S's, is going to be 1. Then the probability of 2 big Y's and a big S and a little s is 1 times 2, so 2. Probability of 2 big Y's, 2 little s's is 1. And the same idea for thinking about these big Y, little y's, right? So I could have 2 big Y, little y, big S, 4 big Y, big S, little s, and 2 big Y, little s. And similarly for the little y's, right? Nothing magical here. Just 1 little y, big S, to 2 little y, big S, to 1 little y, little s. And so let's see, we've got, this is going to be yellow smooth, yellow wrinkled, yellow smooth. So that's what, 4, 5, 6, 7, 8, 9 yellow smooth right here. Now let's see if we can come up with yellow wrinkled. So right here, I've got yellow wrinkled, 3 yellow wrinkled. Then here are my green smooth, and here's my green wrinkled. So I never have to draw a 4x4 Punnett square again. Okay. And so, the idea I've illustrated here is just that the probability of having two independent events, so this is one independent event, that's one independent event, happening together is the multiple of each of those events. Okay, so let's look at now a back cross for two traits. So let's cross that F1 yellow smooth with green wrinkled. So that F1 yellow smooth was heterozygous, right? Big Y, big S, little s. And green wrinkled is going to be homozygous recessive, right? So our green wrinkled genotype is going to be little y, little s. And our yellow smooth will be big Y, big S, little s. Okay, so for this case, again, first thing you need to do is think about your gametes. And let's go back to writing this using the older convention. So we're going to think about all the possible combinations of Y and S. So big Y, big S, big Y, little s, little y, big S, little y, little s, right, for this yellow wrinkled individual, or yellow smooth individual. How about this individual? How many types of gametes? I hope you would say little y, little s. And then you're going to stop writing there because all of the gametes from this individual are going to be exactly the same. So instead of having a Punnett square, all you need to do is draw out a Punnett rectangle. And then let's look at our offspring. So we've got big Y, big S, little s, big Y, little s, little s, little y, big S, little s, little y, little s, little s. So we're going to have a ratio of one individual that is dominant for both of these traits. So yellow smooth, one that's dominant for the first trait, recessive for the second trait, one that's recessive for the first trait, dominant for the second trait, and one that is recessive for both traits, right? Okay. All right, so here actually our genotypic ratio is the same as our phenotypic ratio, right? So 1 to 1 to 1 to 1. And this is an idea that's going to come back later on, as I note here. Okay, now I want you to think back to how we examined some connections between Mendel's first principle of segregation and how that relates to meiosis, right? So that's the idea that the two alleles, when you're considering a single trait, two alleles separate from each other, and offspring inherit just one allele from each of their parents, right? So we saw that that was related to meiosis and to metaphase 1 of meiosis, because during metaphase 1 of meiosis, that's where homologous chromosomes separate. And so if each allele is on a different homologous chromosome and they separate during meiosis 1, then each gamete is only going to wind up with a single allele. So I have kind of the same question for what's going on here when we're thinking about these two traits. And here our finding is that each of these traits seems to be behaving independently of the other trait, right? So we're getting all 4 possible combinations here. And since each of these traits is behaving independently of the other trait, we refer to that as independent assortment. So what that means is these two traits of being yellow or green versus wrinkled or smooth are on different or non-homologous chromosomes. And so the reason why we get all these outcomes is because non-homologous chromosomes line up independently of each other during meiosis 1. So let's take a look at that just to make it very clear. So here, instead of Y, I've used G again, but we can still track this concept, right? So here is a heterozygous individual who's heterozygous for both seed color and seed shape, right? So yellow, smooth. So if so my yellow alleles, this individual is a heterozygous, so they've got a large allele and a small allele on homologous chromosomes, and same thing for smooth and wrinkled. So these chromosomes in meiosis could line up as shown here on the left, and so you could get gametes that have 2 small alleles and 2 large alleles, or they could line up as shown on the right, so that the gametes get 1 large and 1 small allele for each trait. So independent assortment is a physical process, just like the process of allele segregation is a physical process. Okay, so just to restate these again, so the first principle of segregation, is the whole idea that individuals have 2 hereditary factors, or alleles, for a given trait. And these 2 alleles separate from each other during gamete formation. And the physical process that causes that to happen is the separation of homologous chromosomes during meiosis 1. And then again, Mendel's second principle, the principle of independent assortment, is the finding that alleles for different traits move into gametes independently. And I have a special note here that this is only for genes that are found on non-homologous chromosomes, right? So 2 genes each on non-homologous chromosomes. And independent assortment is also a physical process, right? Because the non-homologous chromosomes line up independently of each other during metaphase 1. Okay, so based on these principles, I want to walk you through one more type of practice problem to help you start thinking a little beyond the Punnett square. So the question we're going to walk through right now is, how many different gametes could be produced from individuals with the following genotypes? So the first individual is big A, little a, big B, little b, big C, big D, little d. Okay, so we've gone from a dihybrid cross to a tetrahybrid cross. And please do not try to draw out a Punnett square for this, because there's going to be way too much bookkeeping and you're going to get lost. So we have to tackle this problem differently. And the first thing we have to do when we're tackling this is we need to read the question carefully, make sure we understand. So this question is simply asking, how many different gametes could this individual make? Just the first step before fertilization. So here's how we can think it through. How many different types of gametes are possible for gene A? I hope you would join me in saying 2. How many types of gametes are possible for gene B? Also 2, right? Big B or little b. Are you seeing a pattern? How about C? 2. And D, also 2. Okay, so if it's possible to have big A or little a, big B or little b, big C or little c, big D or little d, then we can have on any of those combinations with each other. So the total number of types of different gametes that could be produced is the product of each of these probabilities, right? So 2 times 2 times 2 times 2, so that's 4, 8, 16. So this individual can produce 16 types of gametes. And now you see why I really don't want you trying to draw out a 16 by 16 Punnett square. So let's look at the second individual. So the second individual is big A, little b, big C, big D, big D. So how many types of gametes can this individual produce? Well, bigger little a, so that's 2. With our little b's, 1, right? Little b is the same as little b, same as little b. 2 for big C, little c. 1 for big D, right? Big D is the same as big D. So this individual can produce a total of 4 types of gametes. So we'll spend some more time in lecture working through other problems that build on this idea, just thinking about these probabilities that I think will help you move a little beyond just thinking about heredity in terms of Punnett squares.